Hello, my name is Matthias Heyden and I'm an assistant professor in the School of Molecular Sciences at Arizona State University. Today I would like to introduce you to some of the research topics in my research group. And from the title you can already tell that we work with computer simulations. Proteins and most other biomolecules are not static objects, but essentially, as you can see in this movie, they wiggle around quite a bit, even in our relatively short simulations. Now, it can be very confusing to look at that, and you have very many different types of dynamics going on. Vibrations, which are pretty fast, picoseconds, maybe nanoseconds, and conformational transitions, which are much slower and sometimes happen within the timescales of our simulations, and sometimes a lot slower on millisecond timescales, which we just cannot simulate yet. These essential dynamics, these collective degrees of freedom which describe the conformational transitions which are often related to biological function, they are the most important part of these dynamics. And we not only try to simulate them in very long simulations, but also we want to learn how to predict them based on the dynamics that we can observe on a short time scale in standard simulations. Another aspect that they're very interested in is solvent-mediated interactions between proteins or other biomolecules. Solvent-mediated interactions are indirect. They are not direct interactions between, in this case, molecules A and B, but they're related to how molecules A and B interact with the surrounding solvent. We can look at that in terms of the difference between the binding energy of these two molecules in solution and, a little bit fictitious, the binding energy in vacuum. And we obviously wouldn't find these two molecules. But if we could measure the binding energy in vacuum, the difference would tell us something about the solvent-mediated interactions. Thermodynamics tells us we can also look at the difference in the solvation-free energy between these two molecules which are separated and the corresponding complex. If you have not heard the term solvent-mediated interaction before, you probably heard about hydrophobic interactions or the hydrophobic effect. And that is just one particular manifestation of such solvent-mediated interactions. Now, if we look at a particular protein complex, like Barney's bar star, then we can now use molecular dynamic simulations not only to quantify solvation energies and their changes when we are pulling the protein complex apart, but with the method that we developed, we can actually generate these three-dimensional maps of local contributions to the solvation energy. That allows us to really basically track down the origins of solvent-mediated interactions between these two proteins. We also like to combine information from simulations on different length scales. For example, in simulations of a single protein in a dilute solution with an explicit solvent, we can simulate conformational fluctuations of the protein as well as its interactions with the surrounding solvent, generating these 3D solvation energy maps. Now we can use that information to construct a model of much more complicated biomolecular systems, for example in a crowded or highly concentrated environment, which actually resembles what it looks like inside of living cells. So simulating that is obviously challenging. We would not try to do that atomistically with all the little details, but we can use the information that we get from simulations of single proteins to generate realistic models of these crowded environments in order to learn how differently do proteins now behave when they're interacting with a multitude of other proteins compared to proteins in dilute solution. In another project, where we also look at multiple length scales, we are interested in liquid-liquid phase separations of intrinsically disordered proteins. This is something that intrinsically disordered proteins are doing in living cells under certain conditions. For example, if a cell is under stress, then they can phase separate into liquid droplets, which basically form now a compartment of their own. They are sometimes also called membraneless organelles. And when the conditions change, they can also dissolve again. Now, in order to understand how this works, we can simulate two intrinsically disordered proteins. We essentially measure the force or the work that we have to perform to pull these two proteins apart, and that tells us something about the effective interaction between two proteins. That, again, we can then use to describe now proteins in a much bigger systems where we reduce each protein to a single particle, because we know how they interact with each other. Now, simulating tens of thousands of proteins, we can, in this case, change the temperature and see how, at one particular temperature, they start forming these phase-separated droplets. The protein concentration is now very high and forming basically their own little compartment, while a few other proteins are left that form now a dilute phase. And if you increase the temperature again, then they dissolve and they form a homogeneous solution. So here in this particular model, we start with a coarse-grained model which still knows about the sequence of these proteins. Every amino acid is represented by a single particle. We generate these effective interaction potentials, and then we can measure the phase behavior of these intrinsically disordered proteins and compare that to experiments. Obviously, to do all these things, we need computers, and we have multiple resources within our research group or shared within ASU. We use multiple programming and scripting languages, visualization tools, analysis programs, many of them we develop on our own, as well as common simulation packages, some of them also developed by us. Many of you may already have experience with programming or high-performance computing. However, this is not a requirement, so if you don't have prior experience, don't let that scare you away. With that, I would like to conclude, and I hope to see you again in our biophysics program. If you have any questions, don't hesitate to reach out. You can reach me here at this email address, mheyden1@asu.edu.